Well, good morning. Up next, we have the Pancreatic Cancer panel. So please join me in welcoming Jean Vidal, moderator.
Well, hello everybody, and welcome to this pancreatic panel session. We're going to have 50 minutes of enjoying time on what is going to be probably one of the challenges that we have for focused ultrasound, which is the pancreas. Before I present my co-panelists here, I want to make two notes. One is, I suppose that every one of you did your homework and you read the presentations that there are. I want to mention them. Irving Koyalen from Virginia Tech University on novel porcine models of pancreatic cancer in histotrypsy. Stephanie Berger from the University of Cologne, MRI-guided hyperthermia in pancreas pig model. Maxime Lafond of LabTau from Lyon, ultrasonic cavitation for controlled pancreas cancer stroma disruption. Jae-Young Lee, that joined us from Seoul University Hospital on FOLFIRINOX plus HIFU in pancreas cancer phase 2 trial. Petros Muratidis, that is joining us virtually from the Institute of Cancer Research in Sutton, London. Histotrypsy and oncolytic viruses in pancreas cancer. And Lucas Sebeck from University of Cologne also on MRI-guided pancreas ablation, HIFU in a pig model. And my last mention is for Tim Mickham from the Focus Ultrasound Foundation that has helped me a lot on organizing this panel. And I wish him fast recovery, and all of us will miss him a lot if he's not here. So let's introduce our panelists. First, I have Jae-Young Lee, professor of the Seoul National University Hospital. Hello.
Hello, I am Jung-Lee. I'm very happy to be here today. Thank you for giving me a chance to present my work. If I summarize my study, my ongoing phase 2 clinical trial is a study to see if the pure mechanical effects of strong but short high-intensity focused ultrasound can be used to increase the effectiveness of anti-cancer drugs. The first conditions we use are very clinical applicable because the patient does not have any discomfort during treatment. Currently, 50 people are enrolled in the 60 registration target. The target population in patients with locally advanced pancreatic cancer or borderline resectable pancreatic cancer who have not previously received chemotherapy for pancreatic cancer. First is implemented as a combination therapy with FOLFIRINOX. Our interim result showed the objective response rate has exceeded 70% so far, so it is encouraging that it shows a higher level than the figures reported so far when using FOLFIRINOX alone. We are also going to see the survival and time to tumor progression, but it is too early to analyze. We are also conducting immunological analysis using blood samples. Thank you.
Thank you. We have here also my friend Eli Blasielewicz from the University of Virginia Tech, Associate Professor of Biomedical Engineering. Eli?
Yes, thank you. So as Sean mentioned, I'm at Virginia Tech, and most of my lab is developing techniques for histotrypsy for targeting a variety of different applications. And a big focus of our work recently, building on the success of some of the liver cancer trials, has been how do we develop this safely, do some of the long-term studies for testing histotrypsy in pancreatic cancer. And so this has involved some both small animal studies as well as some studies in large animals, pig models, both healthy pigs, and more recently we've developed some tumor models to test human pancreatic cancer cell lines in the pancreas. So we're very excited about that work, and we'll talk more about that later. Particularly some of our investigations of the tissue-selective aspects of histotrypsy that we're hoping to see, is that going to translate to pancreatic cancer similar to what we've seen with liver cancer?
Okay, join us virtually. We have Kathy Ferrara from Stanford University, Associate Professor of Radiology of Molecular Imaging. Kathy?
Hi, thank you so much. It's great to have an opportunity to be with you. So our laboratory is working on quite a number of strategies to address pancreatic cancer, including thermal ablation. We've worked on direct thermal ablation quite a bit, and that work is translating that. We also have done quite a bit of work on the incorporation of immune adjuvants into treatment. So either CPG, or we have a paper that will be out very soon on the incorporation of CD40 in treatment strategies for pancreatic cancer. We also have some early data on sonodynamic therapy, and also have been investigating, we've developed a radiotheragnostic approach as well. So lots of different approaches that we're comparing in treatment of pancreatic cancer.
Thank you. And last but not least, Petros Mouratidis from the Institute of Cancer Research in Sutton, UK. Petros?
Yeah, I'm sorry for not being able to be there. I'll just make a general note about where we stand in the pancreatic cancer field as an introduction. It has been a turbulent decade for pancreatic cancer research, in my view. About little more than 10 years ago, we thought that we had cracked the problem of pancreatic cancer, blaming everything on the dense stroma of the pancreatic cancer as the reason why therapies do not work on pancreatic cancer. Then there were a few clinical trials using inhibitors against the dense inhibitors that should limit or deplete the dense stroma of the pancreatic tumors. They didn't really work on most patients, and also in some other patients, the survival outcome was worse compared to the controls. So we started thinking about the dense stroma of the pancreatic tumors instead of thinking of it as something that the tumors create to help their proliferation and their progression. We asked whether it's actually the body that creates this dense stroma to limit the progression of the tumor. And then some more clinical trials targeting hyaluronan show that in patients which had a high content of high molecular weight hyaluronan, using an enzyme that degrades this extracellular matrix constituent, it actually showed some good results, some promising results. And where we stand now is that we think that pancreatic tumors hijack the stroma of the pancreatic tumors at some point to help with tumor progression. And the challenge is to understand not only how to degrade this dense stroma, but also what the cells that are associated with it, like cancer-associated fibroblasts, what they do and what their role is. And this is where my investigations come in. I want to understand how, with therapy ultrasound, what will the modulation of these cells will be, how to increase the immune cells of the microenvironment and make them anti-tumorigenic, and how to increase drug delivery in the tumors as well.
Thank you, Petros. We have a few slides now that we can that will explain a little bit. We can go to the next slide, please. My slides. Okay. So what we have here is just a short overview of how pancreatic cancer has been a challenge and has concerned a lot of interest of researchers, of sites, as you can see in the report of the Focus Ultrasound Foundation recently available. And we see that it concerns a lot of mechanisms of actions and sites that are dedicated to this research. Next, please. We see the treatments the percentage of treatment and indications that we see have been grown. And for instance, in oncological treatments in the last year has been more than half of all the oncological treatments that we have. So pancreas is 45% of the treatments in the indication of focused ultrasound. If we go to the next slide, please. Talking about mechanism of actions for pancreas, almost everything has been tried there. Next, please. We see that in this slide that there is immunostimulation, immunomodulation, there is histotripsy, thermal ablation, tumor destruction, hyperthermia. Anything has been tried there, either for control of the tumor or for control of the pain related to the pancreatic cancer. Next. And the issues that we're going to discuss today, I've been selected these papers that I think that reflect the last knowledge that we have in focused ultrasound for pancreas, and I recommend you that all these papers, and probably the one that Petros has is very comprehensive in this type. And the issues that we have here and that we're going to be discussing is, well, how we guide and target the treatment, the debate between a partial or full ablation to obtain our objectives, how we control the evaluation of the response, the disruption of the tumor microenvironment, how we deal with ducts and vessels, immune enhancement and regulation, the question of drug delivery, nanoparticles, etc. So next and last slide. Here we have the main character of the session today, which is the pancreas. The pancreas is in the very deep of the abdomen, difficult to reach, difficult to treat, very, very sensitive. If you touch it, it responds with pancreatitis, it gets inflamed, and it's surrounded by everything, vessels, ducts, whatever you name it. So let's go for these burning questions. And I have the first one to Jay. Jay, your abstract showed that show significant improvement on the penetration of FOLFIRINOX in advanced pancreatic cancer. Do you have any update on that? Can you illustrate us on that?
Okay. As I just said, it is quite encouraging that the objective response rate is the ratio of patients with partial response or complete response to all patients exceed 70%. Of course, the number is expected to drop slightly as the number of enrolled patients approaches 50. But I think it is still encouraging considering that the objective response rate of the FOLFIRINOX alone reported so far was around 30 to 35%. Just by looking at our hospitals, gastroenterologists, and pancreatic surgeons increasingly wanting to send patients to our research, we expect our treatment to be more effective than existing treatment.
Yeah. And impact of survival and quality of life?
Although 50 people has been registered to the target 60 people, it is difficult to answer at the moment because they are not yet in the stage of discussing survival. I think I will be able to find out next year. In terms of quality of life, there is no change in quality of life because the treatment we do does not cause any side effects during and after treatment.
Okay. And what's next? What are you going to do after that?
Okay. First of all, we are trying for our combined method to be listed in Korean new medical technologies through a randomized trial. If it is registered in Korean new medical technology, we think the patient who have shown effects by our treatment will be able to receive more treatment than number 4. Next, we will find conditions that can make immune boosting. and apply them to patients.
Thank you. Petros, your work is exciting, and it seems that you're still searching for the right approach for this disease. So now you're using histotripsy, and can you tell us why you select that and how it's moving towards treating patients?
Yeah, for the first part of the question, histotripsy, I have chosen histotripsy because I believe, in theory, it should release intact cancer-associated antigens into the microenvironment. So if you think about thermal ablation, there is the danger of heat-fixing cancer-associated antigens, heat-fixing proteins that might be secreted and contribute to immunogenic cell death. For example, HMGB1 that we talked about yesterday, other damage-associated molecular patterns that can help in the induction of the immune response. So thermal ablation might heat fix all that. Whereas histotripsy, on the opposite, by killing cells, will release the cancer-associated antigens, also by permeabilizing cells and bringing them in distress, can release these molecular patterns that might induce an immune response. And in my preliminary experiments, I showed that when I saw that when I used histotripsy, I had more of an induction of immune biomarkers compared to when I used thermal ablation. And also, there is a third reason. Not every lab can have a good, effective MR-guided MR thermometry in their disposal. With cavitation, it's slightly easier to detect it. So we can view it on ultrasound imaging in real time. Also, we have cavitation detectors that we can detect the cavitation. Whereas with thermometry and the heat ablation, that might be a bit more difficult. And there is an element there as well with the thermal dose that is given to the tumors. Different thermal doses will result in different biological effects. And as you know, slight differences in temperature will create huge differences in the thermal dose that is given to the tumor. There is an element of homogeneity there that needs to be discussed. So for these reasons, I ended up working with histotripsy instead of thermal ablation. Low intensity ultrasound, I thought, will not contribute too much to my aim, because my aim was to directly kill the cancer cells. Now, as far as progression to the clinic is concerned, clinical trials of histotripsy are in their infancy, but preclinically the field has exploded now. So this will lead to more clinical studies. And I think until a few months ago, the only published clinical study that I saw was with the VORDRX platform. But now I know that in the UK, there are more clinical studies taking place with targeting liver mets with histotripsy. And also the kidney will soon be targeted as well. And pancreas's turn is coming. So I think large animal models like pig models will be very useful to be able to find the most appropriate acoustic window to the pancreas, and looking forward to see the results of these studies as well.
Thank you. Eli, can you tell us about the large animal work you've done?
Yeah, absolutely. And I think, as Petros mentioned, we're very interested in histotripsy for some of the immune reasons he mentioned, but particularly the big question has been, can we develop ultrasound-guided strategies to deliver histotripsy and take advantage of the high precision, the real-time imaging feedback in some of our large animal models, and then look at the safety profile. So we've done a small number of pigs in a first healthy pig study, looking at 1 week, short time points, haven't seen any signs of things like pancreatitis, pancreatic fistulas, and we're now doing a much larger number of animals with longer term follow-up to further evaluate that. And then in parallel, we've been developing these skid-like pig models that were originally developed at Virginia Tech that allow us to, similar to what we do with our small animal models, grow human tumor cell lines, PDX lines in the pig pancreas, have an actual target to look at. And those results have been very promising. This is a very nice model that we can use to have a target where we can place the tumor in different regions of the pancreas, look at our ability to target and treat. And we've seen, on our first pigs, we've seen we can effectively treat the pancreatic tumors, get a nice ablation zone. And where we're going with this now is obviously doing more studies, but particularly interested in the tissue selective aspects of histotripsy and the safety profile. So in the liver, for instance, we've seen we can ablate liver tumors while preserving large vessels, bile ducts, and get complete ablation up to those structures while preserving them, which is a really unique, I think, feature of the non-thermal ablation modalities, that we can treat near these critical structures. But one of the original questions we heard the comments about the stromal tissue being mentioned is histotripsy going to require a higher dose in pancreatic tumors? We've seen some evidence of that compared to primary liver tumors, but very similar to what we've seen with, let's say, a little bit easier to treat pancreatic tumors at a lower dose than, say, like a cholangiocarcinoma liver tumor. And then comparing that to like the dose that would take to see damage in a vessel, we still see these tissue selective features so far, but we're comparing that with things like different types of bile ducts to understand what the optimal dose is to get a complete ablation but preserve these structures we're interested in.
One of the problems of the pancreas is the bowel blockage, and in the pig, the pancreas of the pig is very difficult because it's between the bowels, hanging from the big for the colon, and it's difficult to reach. And do you think that this can be applied to the human, or your strategies for that can be applied to the human side?
Yeah, no, it was definitely a challenge when we first got started, particularly because we try to fast the pigs and try to follow some of what you would do with your human patients, and they don't listen well, they don't follow our diets. And actually, the abstract you mentioned from our group, led by Dr. Coy Allen, who's our colleague who developed these pig models, he developed his own custard, pig custard that he feeds them now, dosed with some simethicone, gas X, to clear the bowel of gas, and that has worked well from our first studies till now. So he makes that at his house every night and brings it into the pigs. So I don't know if he'll be selling that to people doing these pig studies, but it has been a challenge, and I think that's the question for us, is how do we best prep for our patients. And particularly, we've seen with ultrasound imaging, we can push the bowel out of the way with our handheld probes. But when we have it inside of our therapy transducer coaxial line, we don't have that pressure, so that bowel kind of goes back in place. So strategies for keeping that acoustic window during treatment is going to be important.
In one of the presentations, there is this group of the University of Cologne that also uses this type of filling with, I think, pineapple juice or something like that.
Yeah, we'll have to do I don't know if we've optimized it with the custard, so I don't want to claim that. But.
And also, you've seen that the limitation of treatment guidance with ultrasound is sensitive in the pancreas. And I think you're doing with MRI guidance. So any comment on that?
Yeah, we've done everything so far with pretreatment CTs, but only ultrasound guidance during therapy. And I think one of the big questions that I would have for those who've done more MR-guided procedures is how much of the blockage in the actual therapy path is going to be missed if we're just looking with like a coaxial aligned ultrasound imaging. And we do freehand imaging before procedures, look at that potential acoustic path. But I'd be interested in whether an MR-guided approach is going to be required for a subset of patients, and maybe you can answer that with your experience with ultrasound-guided.
Mm-hmm. Well, in my case, it's been everything ultrasound. So, but I think that ultrasound is good for treatment. So guiding treatment is okay, but targeting probably it's not good enough, and I think we need improvements there. I don't know what you think, Jay. in your case?
Okay, there is, so far there is no problem to target pancreatic cancer, especially head and body cancer with ultrasound-guided hypomachine. The parameter we use is no problem in off-targeting issue because there is no damage on tissues through animal study, and so yeah, we don't think of off-targeting issue is important.
Thank you. Kathy, you have spent a lot of time in the immune treatment of cancer, in particular the abscopal effect, the off-target effect. And in these approaches, combination different therapies, what are your thoughts? How we can optimize the immune response for the pancreas?
Yeah, and I would say that one of the reasons that we've considered thermal ablation, in spite of all the excellent comments that the panel has made, is that if you do a partial ablation, that you do have a hyperemia surrounding that lesion, and that hyperemia can help to bring in therapeutics such as antibodies that can be, I think, instrumental in the response that you're trying to achieve. So it's really that piece that has motivated us to continue to consider a strategy that includes some kind of hyperemia. I do think that there are a lot of opportunities for the use of augmenting drugs to activate the immune system. We know that dendritic cell misfunction is a part of the disease, and therefore adding therapies like CD40 or possibly FLT3 ligand are, in addition to checkpoint inhibitors, can be an important part of the treatment.
Thank you. So to measure the actions on the tumor microenvironment, any ideas on good inflammatory blood markers, immune blood markers? Because the problem in clinical trials is that what we select and when we have to do these measurements.
Yeah, so what we've seen in blood has been markers that are related to type 1 interferons. So if we do some transcriptomics on circulating blood cells, we do see changes, especially if we've given an adjuvant therapy in addition to ablation. And those can be monitored, and they're very similar to, for those who have monitored things like CPG for local treatment and then used blood markers to evaluate treatment. I think the markers that we've seen upregulated in blood are very similar. So they, in part, come from the fact that we're using an adjuvant to enhance the immune system and combining that with HIFU. So yes, I think that can be an important piece. Other pieces that we've been developing at Stanford have been positron emission tomography imaging of immune cells. So we have the ability to track, say, CD8s or other parts of the immune component and evaluate changes in trafficking, also for activated macrophages or changes in activation status. So I think that in the future, that's something that we really want to be able to pair with this kind of focal therapy to be able to assess how the immune system is responding systemically.
Thank you. Petros, any idea of what you're measuring?
I think the main thing that we're all trying to improve is the CD8 cells, the CD8 positive T cells. So unfortunately for immune therapies, there is no marker really to understand if they work or not. But everybody's going for the activated CD8 positive T cell in the tumors. In theory, this is what is gonna do the job down there.
Thank you. Well, the issue of having a full ablation or a partial ablation to obtain an immunological response or to obtain enough to control the tumor. What do you think, Jay? Full ablation, partial ablation?
I can say it's a partial treatment, not ablation, partial treatment in terms of mainly aiming at the deep part of pancreatic cancer. However, the mechanical stimulation that focus beams generate affects all areas of a tumor. In that sense, I can say that our treatment we do is a full treatment. As you know, mechanical stimulation occurs not only at the focus, but also in pre-focusing and post-focusing areas with slightly less intensity.
And Petros, knowing the possibilities of histotripsy of generating more abscopal effects, more immune effects, you in favor of full ablations, partial ablations?
I think whether someone does full or partial ablation, there will always be some residual cells that remain there. So whether they are inside the tumors or in the periphery of the tumors or just outside the tumors, there will always be some residual cells there. So this means that I'm not really sure there is a question mark between full and partial ablation. I don't really see the benefits of partial ablation compared to the full ablation. But there is an issue here. If you can do full ablation, by all means, just go and do full ablation because this will create the maximum intended effect, whether this is growth inhibitor effect or whether it is an immune effect. The full ablation should do it better than the partial one. And there will always be residual cells there, so any combination therapy you use will act on them. But there is a problem here in my view. It might be just from personal experience and it might depend on the model that each person uses. Full ablation, I'm not sure, liquefying the whole tumor, I'm not sure it's possible in some cases. So for small animals like I have, which are really fragile animals, the tumor grows in the pancreas, they are sick, they are tired, they cannot recover that easily from a full ablation with full dose from that. And I assume this will be different in a rat or in a pig and probably different in humans as well. So there is an element of the recovery of the animals when we do full ablation with full dose. There is an element of time. It takes more time to treat a 3-centimeter tumor compared to a tumor that is like 15 or 20 times less in dimensions. And the dose as well high dose of cavitation will create bigger side effects, bigger off-target effects maybe, compared to a lower histotripsy dose. So my suggestion is if you can do full ablation, by all means go and do it. But is it possible? I think each researcher will know for their own experiments.
Thank you. Let's open the question of morbidities when we're treating. Jay, pancreatitis, other morbidities?
Okay. There is a possibility of causing pancreatitis because we use very strong intensity focused beam in our study. But so far there has been no such case, which seems to be because the conditions we use are strong in intensity, but the duty cycle is very low at 1%. However, in the cases of cystic lesions around pancreatic cancer, which can increase the possibility of pancreatitis, are excluded in this study.
Thank you. Eli, what do you think on that?
Yeah, I agree. I mean, I think in our preclinical work, at least, we've seen positive safety profiles. The animals are responding well, and we've been looking for signs of pancreatitis and pancreatic fistulas as potential concerns, but we haven't seen any evidence of that yet. I think we need more data. And as far as I wanted to, Petros made a good point that I think leads to the question of what's the maximum volume we can treat in one session. That's something we're starting to look at in some of our sarcoma and liver tumor work, and I think it's relevant here. So it's not just a question of partial versus full ablation, but when we get to these larger tumors, is there just not only in the small animal models, but if you get to a volume that's too large, should those be applied in fractionated treatments, for instance? That's something we're interested in exploring.
Thank you. Well, with combination therapies, the timing of what is done and when on one of the other, which one is first, is important. Are we to the point where we can say that we know that some order it's important to maintain, and any of those? Kathy, for instance?
Yes, in our work, in our work, it is more important not to completely heat fix the entire tumor, as people were suggesting before. So I think in thermal ablation, it is more critical that there is hyperemia and antigen being generated from the rim of the tumor. But yeah, you're also asking about whether or not there are issues with toxicity that we haven't seen. Yeah, and your last question was,
I couldn't understand your last sentence.
oh, and your final question was, what was your final question for me?
Well, if we have an order established that we say, well, there's something that has to be first than the other.
Yeah, so in our work as well, there has been an order that has been important. So for thermal ablation, the effects that we've seen are so fast that we really feel that we need to have T cells primed and ready and in the draining lymph nodes or in the tumor. And so we've always given checkpoint inhibitors and some adjuvant ahead of time. And that has given us a significantly better response as compared to the reverse order. We've also seen when we do gene sequencing and look at the effect of the order, we've seen that there are changes in apoptotic pathways in the tumor cells such that that upon thermal ablation after something like CPG, we see an upregulation of apoptotic pathways, and we see tumor cells dying from apoptosis rather than thermal necrosis.
Petros, what do you think on that?
Yeah, it's a question that does not have one answer, and it does not have one answer because we are talking about different treatment combinations. So the combined treatment might be an immune treatment or might be, for example, a targeted therapy, a targeted treatment. So if I can start with targeted treatments, there it's logical to do the ultrasound first. Let's say histotripsy, we will treat the tumors with histotripsy. It will upregulate a molecule, a protein, for example, heat shock proteins. Then we can use the heat shock protein inhibitor to target this upregulation. So then in this case, it makes sense to do ultrasound first and then do the targeted treatment straight after that. With immune treatments, with antibody checkpoints, it's different. Although in theory they should also be used to target, let's say, PD-L1, but many tumors express PD-L1 anyway, even without treatment with histotripsy or with thermal ablation. And also have seen in my experiments that when we put an antibody into the animal, even if it is non-specific, so isotype antibodies, it will prime, as Kat said, they will prime the immune system. They will make the immune system more alert. That can be checkpoint antibodies, it can be any other immune adjuvant. So in this case, what I reckon is use a couple of days before the ultrasound treatment, use these immune adjuvants, prime and alert the immune system of the body, and then use the therapy ultrasound to sculpture the tumor and its microenvironment to accept then more immune checkpoints inside it. And there is a third possibility which we must be careful of, which has to do with drugs that are degraded quickly or that are disabled, that might be disabled. For example, CAR T cells. There is an interest in CAR T cells lately in combinations with therapy ultrasound. If we give CAR T cells just after the therapy ultrasound, then with histotripsy, for example, not only we are liquefying the tissue, not only we are killing all the cancer cells, but we are killing all the CAR T cells if they are given with an intratumoral injection. So things like that, we have to be careful, depends on the case, and just use our common sense.
Thank you. Jay?
When I say about the combination of HIF and commercially available anti-cancer drugs, based on the results of our preclinical studies, if a single drug such as gemcitabine is used, I believe it is better to start first treatment as soon as possible after injection of a single drug. In the case of complex drugs such as FOLFIRINOX, I think it is better to start first treatment as soon as possible after the last drug is injected, but the scientific basis is not clear. My logical rationale is that if a concurrent treatment begins before the half-life of all drugs, all effects of various drugs will be enhanced.
Good. Eli?
Yeah, I agree. I think it really depends on what the goal of the therapy is, and if it's enhancing the delivery of the drug, the concurrent delivery is important, or having that drug ahead of time so that therapy can liberate some of the stromal tissue and drive release. But I think good points about the immune.
Okay, the floor is open for questions. If anybody has a question. I have one from the audience, from Khan Mohammed Imran, that says, damage-associated molecular patterns would be self-antigens, signs that indicate a problem if detected. They are not going to be necessarily uniquely expressed between cancer and healthy tissues, and therefore there should be very few T cells that would recognize them. DAMs, therefore, are mainly an innate immune signal rather than antigens themselves to bring the antigen-presented cells in, so the dendritic cells. So my question is, when we are talking about immune modulation, are we talking about pro-inflammation? Kathy?
I think that certainly we do see a pro-inflammatory effect of the ablation strategies that we have used. There is a significant upregulation, for example, of IL-6. But we're hoping to do something that is, with a combination, we're hoping to do something that is able to more subtly change things like dendritic cell functionality by using a combination of immune modulators in addition to the ablation treatment. And in that case, I do think that we can see dendritic cell maturation and activation, for example, which is the kind of thing that we would like to see as we're preparing for the ablation. So I do think that with the right combination of immune modulators, we can do more than just achieve an inflammatory effect. And I think that should be the goal.
Joe, I can also add that the DAMs are just the beginning. So we want to create the innate immunity, withdraw the inflammation, and then try to bridge this to an adaptive response. So it's just the beginning of the DAMs. It's not the only thing we're trying to stimulate.
Thank you. Any comment here? No. There is a question there, here on the left.
Hi, I was just wondering what type of cellular and immuno profiling is done in the current clinical studies to maybe see non-responders versus responders. Like, is there Treg reduction in some, or is there even, novel antigen presentation and novel T cells that weren't there before the treatment, since you're, kind of blowing it up? So, kind of what's been done and what do you plan to do?
In my studies, in my preclinical studies, I have seen an influx of CD8 positive T cells, but not an influx of regulatory T cells or MDSCs. And regarding I think one type of study that is we are lacking in the pancreatic cancer field is the antigen presentation, the functional studies, the cell depletion studies, to show that the activated CD8 T cells that we draw in the tumor microenvironment actually mediate the cancer-specific immune response. These are the studies that we plan to do in the next few years.
Anything else to add? Thank you. There is another question there.
Petros's comment at the end there was really, really important. There's been a lot of work by Bob von der Heide, who's an expert in pancreatic cancer, who shows that CD4 T cells may actually be the dominant T cell type in pancreatic cancer responses, particularly with anti-CD40 stimulation. But my question is pertaining to the earlier discussion about ablation fraction. And so I guess this is really going to go sort of broadly in terms of who responds to this. How much information do we have at the moment about how ablation fraction in the pancreas affects lymphatic drainage and the movement of antigen from the tumors to the draining lymph nodes? And then ultimately, what is the target site for the T cells? Is it the ablated tumor, or are we going to be trying to get those putatively newly activated T cells to target liver metastases? And do our models really allow us to ask that question? Discuss.
Go. Petros.
The lymph node issue is a big issue, which is better to address in a sub-Q mouse model, because there we have clear lymph nodes that drain the tumor. In the orthotopic models that I use, the tumor becomes so big that takes over the lymph nodes. At the end of the treatment, you don't see, not lymph nodes, you don't see a pancreas left. So these are things that we need to understand I need to understand using different models. And it's not a study it's not an answer that I can give yet.
Kathy?
Yeah, so we have looked at both antigen, tumor-specific antigen presentation, as well as specifics of the T cell receptor by using a melanoma model where we have a known antigen, and those were we were able to show specificity in that model. We don't have a similar model for pancreatic cancer in our hands. We are able to look at, if you inject radio-labeled albumin, which is something that we've done in the past, you are able then to watch the albumin, the movement of protein through the lymphatics with PET. And that we found to be very useful. It would be tough probably in an orthotopic mouse model of pancreatic cancer, as Petros was saying, that it would be difficult to see the movement. But in other subcutaneous and in breast cancer models, we've been able to track the antigen that is draining from the tumor using something like a radio-labeled albumin. Is probably one tool that's possible to use. And also, we do have a good tool for activated T cells as well. So there definitely are good PET probes now, both for just T cell numbers, but also for T cell activation.
Thank you. There's another question there.
Thank you. I have a two-part question. First of all, are the antigens that are expressed with histotripsy, sort of hyperthermia or subclinical ablation and ablation similar? And the second part of the question is, what percentage of the antigens are actually at the cell membrane or outside the cell?
Eli?
It might be more for the immune, but I think we've seen an increase in release with histotripsy, and it's mostly outside of the cells after treatment because the cells are fully ablated and into an acellular liquid homogenate. So I don't know about, maybe if Petros wants to comment on any histotripsy versus HIFU in his models. But
Petros?
The pancreatic tumors are low in these cancer-associated antigens. So it's very difficult, if not, it's very, very difficult to detect specific ones. We want antigens that will be expressed on the surface. It won't do if they are expressed inside. So in order to be able to target metastasis, for example, we need them on the surface. The experiments we will do is to express exogenous antigens inside the cancer cells, treat them with histotripsy, and then see if they are released and if they activate the immune response. So there are experiments to be done. There is a very interesting study that has done this thing in another model with the GP33 antigen, so we will do something very similar.
Thank you. We have a couple of minutes for the last question.
Okay, yeah, thank you. I'm one of the guys from Cologne. We're doing this HIFU ablation under MR guidance. I wanted to give a brief comment because you asked before about the avoidance of abdominal organs. So under MR guidance, we were able to do our PICs. We did 13 in our follow-up study. We were able to avoid abdominal organs. The ablations on other organs that we saw were mostly associated with the small size of the pancreas, so that we were not able to really contain the ablation cell inside the pancreas itself, because we don't have a sophisticated tumor model like you have, so just very thin pancreas. On that note, because we were working under MR guidance and are also working on getting really the hypothermia going, maybe also to induce localized drug delivery at some point, I wanted to ask what your perspective is. Do you think that has a future, the targeted drug delivery, or is that too complicated in a clinical setting?
Eli?
Yeah, I think it's a great point about the first of all, on the healthy pancreas. You know, we've seen the same thing, like testing in the healthy pig pancreas, very small target, hard to do our volumetric ablations and accurately assess for safety without hitting some of the other organs around it. So we've seen similar things. To moving to the tumor models has helped. I can't comment on the specifics of the data, but another focused ultrasound company has that I'm not involved with this, has used the pig models at Virginia Tech and for that focus on drug delivery, and they've seen some promising results that they'll be publishing. So I think there is an application there for enhancing bioavailability of these drugs for pancreatic cancer. I don't know on the clinical side, I can't comment on that. But.
Thank you. Well, I think we will run out of time, but I want to have one last question, which is the impact on resectability. I don't know if we can, and the floor is open for responses of all your panelists. And do you think that we can impact on resectability of the pancreatic tumor if we do an ablation? Have you had in your patients a patient that you have reduced the tumor in order to make it resectable?
I am also analyzing the resectability, surgical resection rate after our combined treatment. Interim result says some positive result, but yeah, it is too early to say that. However, objective response rate increase means the more patient shows partial response or complete response. So it is related to the increase in surgical resectability. We have to find out in later.
Thank you. And Petrus and Eli, you that work with histotripsy, do you think that given the tissue selectivity of histotripsy, you could treat the tumor into the vessels and dissect the vessels, get rid of the tumor out of the vessels, and make the patient resectable?
Generally speaking, I think the issue of resectability is something we have not thought about enough. I think there is an application there for therapy ultrasound and histotripsy to make borderline resectable tumors resectable. As the technology progresses, we can go into the details and see how we can detach it from vessels. So yeah, it's just something that me personally, I have not thought about. But there is also an issue of whether I have spoken with a few surgeons and they all tell me the same thing. Even if they spend hours doing resecting the tumor, the pancreatic tumor, the problem is still there. They cannot scoop up all the residual cells. The pancreatic cancer will come back again.
Eli, any thoughts?
Yeah, I think specifically around the vessels, I believe we'll be able to do that based on our early data showing that you can ablate, liquefy right up to the edge of the vessel and treat. I think the question for like a treat and resect is, again, that full versus partial ablation. If you're fully liquefying that tumor, you're probably going to want to wait before you're scooping that up out of there and give it some time before you move to resection.
Mm-hmm. Well, thank you. Thank you, the audience, for being with us, and thank you, the panelists. And I think we're ready for the break.
Well, thank you very much. We do have a short break now. We're going to ask that you be back at 10:20. A general reminder for young investigators right now, we're going to take a photo on the back stairs near the escalator. So young investigators, photo op right now. Please be back at 10:20.